Hello, and welcome to our second of 5 fully vodcasted lectures. If you're watching this in the lecture hall right now, then of course it's Friday the 20th. And a few things up and coming to remember. We do have our second homework assignment due on the 24th, and it's just really important to get going on this homework assignment earlier. It is a little bit harder than the first homework assignment. It's also got more questions, so you may want to delve into it maybe a little bit earlier than you did last time. I know some of you probably did it very early last time, but it's not one that you want to put out to the wayside. And if you do need any help with it, please let me know. I will be having my office hours on Monday night and Tuesday night. I'll be available in lab on Tuesday, as always. And also today, Friday, one of my TAs will be available in open lab from 12 to 2 to help out. And in fact, he's a graduate assistant and is capable of helping you with any of your questions that you might have on macromolecules. And towards those ends, actually, I did post all of the answers to the game questions that we worked on Wednesday, so you can find those on either eCompanion. You can also find them on the web page, so whatever works easiest for you. Please do think about emailing me with your group members' names as well as your tentative topic. I know that some of you have already done so, and some of you have given me your group names, but you haven't talked about a topic yet. So wherever you are, please just let me know and keep in touch, and hopefully we'll get those nailed down within the next couple of days. We do have a pre-lab due prior to the lab on Tuesday, and we'll be performing lab 2 on that day. So I'll be stoked to see all of you for that lab on Tuesday as well. Let's jump in. We've got a lot of exciting things to talk about, and if you remember last time, we hadn't quite had a chance to get to the last category of macromolecules. These are the lipids, and just as we started with the carbohydrates and the proteins, let's start by listing 2 functions of lipids, and in fact, I think I've got a list of more than that. Now, one that might come to mind right away is that in fact lipids are fats, so fats can be an incredibly efficient fuel source. So fats are in fact a source of energy. They can also play many other roles, some of which may have come to your attention and others maybe not. Maybe some of you immediately thought of phospholipids, which of course play their role in creating biological membranes, and we're going to talk a lot more about that today. They also, as we mentioned, can be a great source of energy. In fact, fats are packed with a lot of power. Fats are just full of energy. They can also play a role in thermal insulation and padding. And I know that that's not necessarily a function that we celebrate a lot, but I have a picture here from one of my prior students. This is Marcy Trana, and she actually worked in Antarctica with polar bears. And you can see here she's comparing her paw size to the size of a polar bear paw. But what I wanted to talk about was the pads within the polar bear's paw that allow it to have a lot of thermal insulation and padding, and those are full of fats that can change in their fluidity depending upon the temperature outside. They can become more fluid with cold temperatures to enable a sort of thermal insulation and padding function. So surface protection, we can talk about what waxes can do. Waxes are a type of lipid and how they can protect the surfaces in many cases. Cell signaling and recognition, certainly fats can play a role there, or lipids as a whole can play a role in that function as well. So there's just a lot of diverse ways in which lipids play a role within a cellular system. And let's go ahead and look at the categories, if you will, of lipids just briefly, talking about the fact that there is more than one category. We have our classic fats, which are also known as triacylglycerols. Of course, we have our phospholipids, which make up or comprise a great percentage of biological membranes. Steroids and waxes are also categories. But in this picture, we're seeing a picture of a fat, a triacylglycerol. And what we recognize is that this molecule has a glycerol backbone. Let me go ahead and point out that glycerol backbone. That's this region here. And attached onto that glycerol backbone by an ester linkage, Della maybe recognizes those, right? You're our chemistry expert. So we recognize that being a long chain of carbons attached on to that glycerol backbone. So we can recognize that these are the fatty acyl side chains. And for those of you who don't speak a lot of chemistry, just a quick note that all of these here are hydrocarbons. So if we were to actually write out what was there, it would be carbon attached to hydrogen in each one of these positions. So we'll look at this. We've got a lot of carbons in the side chain, starting up here with carbon number 1 and numbering down. We have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14 carbons. Wow, that is a long hydrocarbon chain. And if everyone remembers how hydrocarbons feel about water, they really don't love it, right? Fats are not miscible with water. This is a highly, very, very in fact, you know what? I've got a glass of water over here, and I just happen to have a little bit of vegetable oil, and we can remember that, of course, fats and water don't mix. All those hydrocarbons, definitely not mixing well with the water, right? Separating out. And so the fats hate water. And in fact, fats in our body and in a cellular system, in any cellular system, will be stored in what we call an anhydrous form, meaning that they're stored very much cordoned off from water. And that actually makes fats kind of an efficient fuel because they're an incredibly lightweight fuel because they're not saturated by water, right? Think about it. When you go backpacking, you want to take anhydrous foods, right? Those that are dehydrated and not associated with water because they're lightweight to carry around, right? And fats are a nice, lightweight, highly packed with power form of energy. Versus carbohydrates, right? Carbohydrates are the awesomest source of energy, but remember how heavy they are because they are incredibly hydrophilic. They're so water loving. And in fact, hey, I just happen to have some cornstarch over here that I've been throwing around my office and getting a mess around. But let me go ahead and add it to some water. And I've got another cup of water here where we can see that, in fact, a carbohydrate is going to be highly miscible with water. So we can kind of stir that around. I added some of that cornstarch and we see it very much go into solution. Oh my gosh, let's write it down again. Like dissolves like, right? So remembering like dissolves like, where those carbohydrates are of course very much like water, whereas the fats are very nonpolar and do not like water. So that comes into play with our fats as well, comparing and contrasting the carbohydrate macromolecule group with the fat or the lipid macromolecule group that is very water hating. Now, the question here asks something a little bit different. Why are these highly hydrophobic molecules very, very efficient at energy storage? Now, it might be that Candace and Britt and you other nutrition geeks out there, you might be thinking, oh, I know why a fat is so packed with power. Notice that it has so many hydrocarbons, right? Each one of these is a hydrocarbon at each one of these positions. So this is full of reduced carbons. That is, carbons bound to hydrogens. They are highly reduced. So when we eat and we take in a fat, we can rip and strip high energy electrons off of that fat. We can carry those high energy electrons to our electron transport chain where they're passed from one member to the next member to the next, and that energy is used to pump protons, and that proton gradient can be used to make, you got it, ATP, baby, right? So this is a really big take-home message. Things that are highly reduced, that have lots of high energy carbon-carbon and carbon-hydrogen bonds, are wicked high in energy. And that's why a fat is not only an efficient storage molecule, right, lightweight and anhydrous, but it's also a high energy storage molecule, lots of energy in there, because the bonds are highly reduced carbon bonds, lots of carbon-hydrogen bonds. Okay, let's go on and take a quick moment to look at the phospholipids. and talk about the difference between a fat, a triacylglycerol, and a phospholipid. Notice that the difference occurs at carbon number 3. Carbon number 3, instead of being bound to another long fatty acyl side chain, instead carbon number 3 is bound up here to a very polar alcohol with a very polar phosphate group. So what we see is what we might term, and I bet you remember this, what is it, that word that means to have a dual nature? Right? Something with a dual nature. Remember we had our alpha helix that was amphipathic? So amphipathic, this is an amphipathic molecule, right? It has a very water-loving head, but two very, very water-hating tails. So that's what we see in a phospholipid. That's a horrible circle. Two very water-hating side chains, as well as a water-loving polar head group. Okay, great. So this is a phospholipid. This is how a phospholipid differs than a fat. And we can recognize how phospholipids can sort of form the backbone of biological membranes, because phospholipids can come together in 2 layers where we get the polar head groups facing outwards and the long fatty acyl side chains facing inwards, cowering away from water. And so in a phospholipid bilayer, this is the kind of orientation we'll see the phospholipids taking, where the very polar head groups will face either outwards, this is out here, facing the aqueous exterior of a cell, or inwards, facing the aqueous interior, where the hydrophobic side chains will cower inside. I've got a picture of that on the next slide, showing a phospholipid bilayer. And I want to note that sometimes when I talk about a bilayer, I'll talk about the outer leaflet and the inner leaflet. So recognize that this is the outer leaflet. Okay, so this area here is the outer leaflet. And this in here, this is the inner leaflet. Together, they come together to make that phospholipid bilayer. So as we go into our next coverage, which is regarding this prokaryotic cell structure, one of the things that we're definitely going to focus in on is membranes. So the phospholipid bilayer, you could think of it as the cornerstone of the membrane. We know that there are some differences because membranes, of course, have proteins in them and other molecules that we'll introduce here in a moment. But the phospholipid bilayer is sort of the cornerstone of the cellular membrane. Okay, here we go, onwards and upwards into our next set of coverage, which really literally means that we're leaving macromolecules behind. It's a little bit depressing, but since I was kind of feeling bummed about it, I thought let's do one more review question. So I've got a review question for you. A sweet-looking protein. It's a component of the flagella of a bacterium called Vibrio parahemolyticus, and this is a really fairly newly added structure to the protein databank, meaning that they've only fairly recently determined the structure of this protein and been able to show a cartoon depiction of it. So we have a question just helping us review about proteins. So let's see if we can do a good job of answering it. All of the following accurately describe this protein except. Okay, so A, this protein is characterized by alpha helical secondary structure. B, the subunits of this protein are linked by peptide bonds. C, the amino acids valine, leucine, and phenylalanine would likely be found on the side of the red helix that faces the interior of the protein, and that's this one here. This protein likely has very high proline content, and this protein has quaternary structure. So notice that we're looking for the exception here, so we're looking actually for the false statement. If we return up to A, we recognize that as being a true statement, so I'm just going to put a little true here, because obviously there's a lot of alpha helix in this protein. The subunits of this protein are linked by peptide bonds. Of course they are, right? They're amino acids all linked by peptide bonds, so we can say that's also true. The amino acids valine, leucine, and phenylalanine, well, Roman would tell us, just as he did last time, that phenylalanine is a very, very water-hating amino acid. So it's likely to be found in the hydrophobic interior. So that sounds like a true statement. This protein likely has a very high proline content. Wait a minute. Proline? Isn't that the one that induces the kinks, right? It's the kinky amino acid. So we'd be likely to see proline right here where there's a direction change. We might see it in a couple other places, like maybe here where there's a direction change. But for the most part, proline is not very common in alpha helices. In fact, we call it a helix breaker because it likes to induce kinks. And kinks don't go very well in the nice, smooth, swooping turns of an alpha helix, right? So we generally would say no to that answer. So let's call that false. This is going to be our answer, isn't it? But let's see why this is true. This protein has quaternary structure. It does. It has more than one polypeptide chain. But I bet you're thinking, how do you know? I mean, like you're looking at that picture and it kind of looks like whatever. I don't know. So let's see. How you know is by finding more than two ends. If there's only one polypeptide chain, there should only be two ends, right? But if there's more than one polypeptide chain, there should be more than one end. So let's just circle the ends. I see one here. I see another one there. I see a third there. Already we've counted up above 2, so we know this must have some quaternary structure. Hey, look, I see the fourth right there. Okay, so this definitely has quaternary structure. Marvelous. That was a phenomenal review. Let's go on and begin our conversation regarding the prokaryotic cell structure, and we're going to zone in and kind of focus in particularly on bacterial cells. But we'll also talk a bit about Archaea, and to begin our whole survey, I want to make sure that we have the big picture view before we go and delve into the cellular structure. So let's begin by noting that all life, no matter the life form, can be categorized into one of 3 different domains. So looking at these 3 domains, we have the first that perhaps is the nearest and dearest to us, eukarya, and of course that's the one that includes us. It also includes all of those crazy protists that you saw swimming around in the pond water yesterday. So all of those, what we might term protozoa and algae that we saw on our slides, those would also fit into the world of eukarya. Now, the other domain, it's very separate from the other 2 that we're going to talk about, is they're called the Archaea. Now this one's going to be Laura's favorite because she likes the extremophiles that live in Yellowstone. And to date, the Archaea that we have characterized, many of them tend to be the extremophilic kind. They live in incredibly high temperatures or incredibly saline environments or very, very low pH environments. So by our estimation, perhaps a little bit egocentric, we see their environments as being very, very extreme. Let's talk, though, last but certainly not least, in fact, quite the opposite, most would be the more accurate way to say this. Bacteria by far outnumber everything else. There are more bacteria than Archaea and eukarya combined by far. Bacteria are by far the dominant life form on Earth. And in fact, I have a passage that I want to read to you, and I thought Adam would enjoy this with his love of poetry, and I suppose probably a love of beautiful writing. So if you haven't yet, you should check out Stephen Jay Gould. He writes really great stuff that is both, I think it's both artsy and sciencey. So let me read you a passage from some of his writing from a piece called The Planet of Bacteria. On any possible, reasonable, or fair criterion, bacteria are, and always have been, the dominant forms of life on Earth. Our failure to grasp this most evident of biological facts arises in part from the blindness of our arrogance, but also, in large measure, as an effect of scale. We are so accustomed to viewing phenomena of our scale, sizes measured in feet and ages in decades, as typical of nature. Individual bacteria lie beneath our vision and may live no longer than the time I take to eat lunch or my grandfather spent smoking his evening cigar. But then, who knows? To a bacterium, human bodies might appear as widely dispersed, effectively eternal, or at least geological, massive mountains fit for all forms of exploitation. Planet of the Bacteria, Stephen Jay Gould. So bacteria are, by far, the dominant life forms on Earth. Now, if we want to, and some do, we can consider bacteria and archaea together to get a term that I bet you've heard of called prokaryotes. Now, prokaryotes can be spelled with either a C or a K. Here I have it spelled with a K. Occasionally I may spell it with a C. Old habits are hard to break. But I want to bring up something kind of wanky. I want you to think about whether or not the term prokaryote is even warranted. Or does it really just say not one of us? In fact, there are many great scientists right now who are arguing that the group prokaryote isn't really warranted because archaea are not more related to bacteria than they are to eukarya. So really, this is something throughout the rest of the semester that as we learn more and more about bacteria and more about archaea and more about the eukaryotic microorganisms, I want you to think about whether we should still have and hold on to the term prokaryote. As we move into our coverage, though, we're going to focus more on bacteria now than any of the other domains of life. And I want to begin by talking about what we call the binomial system of nomenclature. This is a two-word naming system. And essentially what it's like bacteria have a first name and a last name. But on a more scientific level, they have a genus name and a species name. So when we write their genus name and species name, it's always italicized or underlined. Most of the time we italicize it, but when I write it on the board in lab, for example, I'm going to underline it because that's a lot easier. Either way is totally fine. The genus name is always the first name. It's what comes first. Notice here that the most famous bacterium, Escherichia coli, Escherichia is the genus name. Notice that the genus name is uppercase, right? It's capitalized. And the genus name comes from often, historically, it often comes from the name of the discoverer. Anymore, that's not so much what we see. But Theodore, a man named Theodore Escherichia, is the one who discovered E. coli. So that's where that name originates. Coli, this one makes more sense. The species name, notice that it's lowercase, and that it comes from where that organism is isolated, from wherever that organism is found. So you might recognize that Escherichia coli was isolated from the colon, and it is in fact a very dominant bacterial normal flora within our gastrointestinal tract. So it comes from where it is isolated, the colon or the gut, right? The lower intestine. So the genus name is always written out the first time you use it, but then if you want to use it subsequent times, we get a little lazy and we start abbreviating it. So Escherichia, after you've used it once, we would start writing it as simply E. And in fact, you've probably heard more of E. coli than anything else. So E. coli is the abbreviated form that can be when you go to write your lab reports, the first time you use the name, you're going to want to write it out. The second time, it's okay to abbreviate it this way. Now, there's one last portion of nomenclature that's important for us to review, and that is that we not only talk about genus and species names, but we can also talk about strain designation, which really does matter. So in lab, we work a lot with something called E. coli K12. It is a non-pathogenic laboratory strain. That is, it's not going to make you sick. But we've also heard of things like E. coli 0157H7. That's a gnarly food pathogen. So the strain designation really does matter. E. coli K12, non-pathogenic. E. coli 0157H7, very pathogenic. I'm going to jot down E. coli 0157H7 because probably some of you remember certain outbreaks of that particular food pathogen. And the strain designation means a lot. So it's E. coli 0157:H7, and that is the strain designation. Now we might want to go ahead and make sure that we underline E. coli, since it's not clear whether that's italicized. Marvelous. Let's go on then from talking about nomenclature, which I promise this is something you're going to need to put it onto your long-term memory, because this is definitely something that you're going to need to reuse as you write lab reports and whatnot. But now we can talk a little bit about cell shapes, that is overall morphology of cells. Morphology means the form of a cell. That is shape, arrangement, and the general form.